diagnosis. Without getting proper diagnosis, left, right, and center administration of the antibiotic is something deadly. This means probably we will end up individual having a huge amount of antimicrobial resistance in his or her normal flora. Okay, I hope you may be knowing what is normal flora. Normal flora are the commensal organism which normally stays in an healthy individual tissue like intestine, oral cavity, nasal cavity, okay, as well as on the skin. Okay, they survive usually and normally. Okay, if you take unnecessary antibiotic, then it will end up having antimicrobial resistance in those normal flora as well. And this is a very important fact. For any viral infection, antibiotics do not work. Antibiotics are basically meant to work against bacterial infections. Okay, they are either product of actinomycetes or fungus, but they are meant to act against bacteria, not the viruses. So in common flu or viral infection, we should not administer antibiotic. WHO Global Health says World Antibiotic Awareness Week 12 to 18 November 2018. And in this year, they have celebrated this Global Antibiotic Awareness Week. And they also pulled many conclusions which were later on published in 2019. And they had also supported, and they have also advised and recommended certain practices which can prevent the antimicrobial resistance to a better extent, right? Because what is the challenge nowadays is we are almost on the end of antibiotic pipeline. Now, no more new antibiotics are developing or under the research mode, which can be of use to any individual in future. So today or tomorrow, we are going to have no antibiotic situation with us. And what shall we do in that kind of time that we all don't know? Okay. So aim is to increase global awareness of the antibiotic resistance, to encourage best practices among the general public, healthcare worker, policymakers, and to avoid the further emergence and spread of the antibiotic resistance. That was the aim of that week celebration. Change can't wait. Our time with the antibiotics is running out. As I've told you before, now there is no new antibiotic coming by any of the pharma company. Reason being, the antibiotic research is very expensive and extremely time consuming. A new antibiotic takes at least 15 to 20 years to come into the market after laboratory based research, then toxicology studies, then animal trials, then human trials. So they take this much duration to come into the market. Okay, antibiotic resistance poses a big threat to the global health, because whatever antibiotic we discover, sooner or later they become resistant in most of the medically important bacteria. And that's how we are left with no use of that particular antibiotic or no productivity of that particular antibiotic in any kind of infection. So that is the biggest problem with antibiotic. And in other words, or in other scenario, if we see the other viewpoint, the microorganisms are very much smarter enough when they come across any compound which we discover, and they find out a cellular bypass mechanism where the antibiotic cannot alter their growth and reproduction. Antibiotic resistance can affect people of any age and in any country. There is no bar. There is nobody who is discounted or nobody who is exempted from this challenge. Everybody poses the same threat. Drug resistance is basically started from where? Drug resistance is circulating amongst human, animal, and the environment. Unjudicious or uncontrolled, irrational use of antibiotic has led us to this situation where the animal feed is being treated with antibiotic. That was a very old practice. Then irrational use of antibiotics in humans for a very small illness. General practitioners and people used to prescribe antibiotics, and that too, even I have seen many examples of higher standard, higher antibiotic, okay, which are considered to be the last resort, they are prescribed on a simple OPD based stable patient. Whereas actually those antibiotics like carbapenems, cholesteins, and other antibiotics like our what can we say, this linezolid for gram-positive bacteria, vancomycin for gram-positive bacteria. These antibiotics are actually reserved drug. They are used only in the last resort whenever the infection is not at all being controlled by any other antibiotic, and you are left with only these options. At that time only they are supposed to be used. But actually people uses this antibiotic irrationally in even a simple illness. So that is the problem and that is the drawback. Uh, so far, laboratories worldwide are taking actions by tracking the spread of this drug resistance at microbiology lab level. The tracking and recording and reporting of this antimicrobial resistance is somehow in practice. Additionally, there has been a new practice which was suggested in 2019 by WHO is restricted reporting. Say, for example, in a gram-positive organism, we usually test 13 different antibiotic whenever we test for antimicrobial susceptibility test. Now, while we test this 13 different antibiotic, out of this 13, 3 are higher antibiotics. So we should restrict those antibiotic reporting. We should not report those antibiotics at all in an culture and susceptibility report. If the rest 10 or out of those rest 10, you have got few antibiotics sensitive. Okay, I hope you are getting my point, but more you will be clear until you will be probably whenever you will be coming into the laboratory posting and whenever you will be given a hands-on practical of antimicrobial drug susceptibility test and resistance mechanism understanding. At that time you will be taught for this. But a reserved drug need not to be reported if the non-reserved or unreserved drugs are susceptible. That is a new practice started, and that probably many of the microbiology lab is practicing nowadays. And one most important fact is before consuming any antibiotic, please contact the authorized practitioner, general physician, whether the indication of antimicrobial treatment in that particular patient are fulfilling or not. Okay. And another bad practice is never share the antibiotic to anybody, which we all may be knowingly or unknowingly doing very frequently and easily. That is not a good idea to share antibiotic of one particular patient to another patient. So effective waste treatment can also protect the environment and reduce the antimicrobial resistance. Waste here I am targeting the animal dung or animal excreta. Animal excreta, because of heavy use of antibiotic in animal feed, is also containing the organisms which are having antimicrobial resistance normal flora. A proper waste treatment will also restrict the spread of that antimicrobial resistant genes. Okay. Now the global action plans on antimicrobial resistance says get informed, gather data, prevent infection, regulate the medicines, invest now. Our time with the antibiotic is running out and change can't wait. So that was the preamble of sensitizing you for the reason behind teaching antimicrobial agents and antimicrobial resistance and how big the problem is. Okay, so let me just take you through our lecture of antimicrobial agents. Who is he? Anybody? Anyone knows him? Who is this fellow visible in this picture? You must have seen the photograph. Yes, Alexander Fleming. He is Alexander Fleming. He is the individual who discovered the dramatic compound. First ever he discovered antibiotic penicillin in 1940. And that antibiotic during the Second World War has worked as life-saving medicine. It has worked on lot of soldiers who were wounded, who were having septic wound, and their lives have been saved by penicillin. So Fleming discovered penicillin in the petri dish because of contamination of the penicillium. Okay, and then he could identify the compound. He filtered that compound which was secreted from the fungus, and it was preventing the growth of other gram-positive bacteria like Staphylococcus. So this would remain our learning objective for the session. Describe, define sources and classification of antimicrobial agent. Describe mechanism of action of various antimicrobial agent. Describe the method of testing bacteria for antimicrobial sensitivity, disk diffusion, and MIC. Explain antimicrobial resistance with example, and antibiotic stewardship program. What is that? That I will brief you. So antimicrobial drug, 1928, Fleming discovered penicillin, which is produced by penicillium. In 1940, Howard Florey and Ernest Chain performed first clinical trial of the penicillin, and which has helped everybody. An antimicrobial agent is a substance of natural, synthetic, or semi-synthetic origin that kills or inhibits the growth of microorganism but causes little or no host damage. Okay, it causes no damage, or either it causes, then it is a little damage, which in the pharmacological term we normally know it as adverse drug reaction. Okay, an antibiotic is a substance produced by the microorganism that at low concentration inhibit or kill other microorganisms. Okay, so this is the natural one, and from this natural one we have received, or we have researched on it, and humans have nowadays derived more than 300 different compounds. We nowadays have more than 300 different antibiotic compounds with more than 17 different classes of antibiotic which can be used in humans. Just you can imagine, just in the journey of 80 years we could reach to huge number of antibiotics. So these are few examples of organisms producing the substance, and which has been used in the form of a antibiotic. A gram-positive rod or bacillus subtilis produces bacitracin, Bacillus polymixa produces polymyxin, various actinomycetes producing various antifungals and antibacterial, and various fungi producing antibacterial and antifungal. So we have used this substance as natural one, and we have either altered their chemical structure a little, and we may have derived a new particular congener or a variant of that particular class. So we have nowadays more than 300 compounds available with us. So broadly explaining you the classification of antibiotics based on the mode of the action, they are either bacteriostatic or bactericidal. Bacteriostatic means they inhibit the growth of organisms, and bactericidal means they kill the particular bacteria. So that is based on their mode of action and capability to act. Based on the spectrum of activity, we have either broad spectrum, which is effectively working against lot many varieties of bacteria, or else we can have narrow spectrum antibiotics. These narrow spectrum antibiotics used to work limited or a very selected class of organisms, bacteria or fungi, or even sometimes viruses. So they could be either narrow spectrum or broad spectrum, and they could be either bacteriostatic or bactericidal. So this is the spectrum of action for selected antimicrobial agents on prokaryotes, eukaryotes, and viruses. The spectrum of activity of selected antimicrobial drugs is something like these. For prokaryotes, largely we are targeting bacteria, and the maximum amount of antibiotics are also available against bacteria, because till date the larger class of infectious agent were considered to be bacteria. Later on we could identify fungus and parasite as important pathogen. Later on we could see that viruses are also creating problem. Previously it was understood that the virus can be very well handled by our innate immune system, or there may be at least a day or two or a week suffering in the viral infection, but antibiotics are not always necessary, or antivirals are not always necessary. But nowadays there are certain complicated and challenging viral infections are also there, which also need the antimicrobial or antiviral treatment. Okay, but no antibiotic till date we have got which is effective against all kind of microorganisms. We really do not have any single compound which is effective against huge number of organisms or huge varieties of organisms. But we may have a broad spectrum antibiotic which may be working against one or two or three classes of bacteria, say for example as erythromycin, tetracycline and sulfonamides, which is effective against gram negative, gram positive and chlamydia rickettsia as well. Okay, so that is at a glance about the antimicrobial agents. Now let me just tell you the actions of antimicrobial drugs. So this is a diagram of an typical or idealized bacterial cell, and it is describing the important physiological activity which is going on within the cell. So principal thing is DNA replication, which is taking part as a primary process of cell division. We can also have transcription, which will derive mRNA from the DNA genetic material of the bacteria. So that is producing mRNA. We can have proteins out of mRNA with the procedure of translation. That is also delivered by certain enzymatic activity. Okay, so we can have antibiotics which could be working on cell wall synthesis or which could be preventing cell wall synthesis. The antibiotic which is stopping or inhibiting the cell wall synthesis are penicillins, cephalosporins, bacitracins, and vancomycin. Antibiotics inhibiting the protein synthesis at the translation level are chloramphenicol, erythromycin, tetracycline, and streptomycin. Antibiotics causing harm or permanent injury in the plasma membrane or cell membrane is polymyxin B. And the another one mechanism where inhibition of the nucleic acid replication and transcription, which is also very crucial for cell division and cell metabolism. The antibiotics working there are quinolones, fluoroquinolones, starting from ciprofloxacin, norfloxacin, ofloxacin, levofloxacin, and rifampicin. Okay, and the last one, inhibition of the synthesis of essential metabolite, that is sulfonamides and trimethoprim. Okay, so this is the normal places where the antibiotics work, and they ultimately lead to either inhibition or death of the bacterial cell. I'm just stopping here and just asking you, is it clear up till what we have discussed with everyone, or shall I repeat anything? If anybody wants to revise anything or want me to re-explain, please text, please chat, please write in chat with the particular topic if anybody wants, or else I'll wait for 10 seconds and I'll go ahead. Okay, I shall go ahead. So these are the mechanisms which we have already discussed. Okay. Inhibition of the cell wall synthesis, the mechanism number one and most commonly used, and large number of antimicrobial group are belonging to this class, is beta-lactam group, in which the penicillins, cephalosporins, carbapenems, and others are members. Okay, and recently added one is the glycopeptides. Recently added one are glycopeptides. Other inhibitors of the cell wall synthesis are antimicrobial or antibiotic effective against Mycobacterium tuberculosis and other mycobacteria. Interfere with the mycolic acid synthesis or incorporation of this antibiotic as one of the key subunit of mycolic acid, and then stopping the cell division at that point, that is isoniazide and ethambutol. Okay, so that is one. Inhibitor of protein synthesis. Inhibitor of protein synthesis are We have got aminoglycosides, we have got macrolides, we have got chloramphenicol, we have got lincosamides, we have got oxazolidinediones, we have got streptogramins, we have got tetracyclines. Okay, these are working on one or the other places of the uh, protein synthesis that is happening at the ribosome level, and they might be attaching with 30s or 50s subunit of ribosome, and they are preventing the uh, protein synthesis, and that's how they hamper the cellular metabolism, and the bacteria die with this. So that is example, or these are the classes which is working against protein synthesis in bacteria. Inhibitors of nucleic acid synthesis, inhibitors of RNA synthesis, and inhibitors of DNA synthesis. In inhibitors of RNA synthesis, we have got rifampicin, rifamycin, rifampicin, rifampin, rifabutine. These all are working only and only against bacteria. Okay, so they are antibacterial, and to some extent they have got anti-tubercular activity. Inhibitors of DNA synthesis, we have got fluoroquinolones. They are again the antibiotics considered to be active against bacteria and tuberculosis. Nalidixic acid, ciprofloxacin, ofloxacin, norfloxacin, levofloxacin, lomefloxacin, sparfloxacin, and getifloxacin. Antimetabolite antimicrobials. Antimetabolite antimicrobials are the antibiotics which has got structure which is resembling to the structure of substrate of one particular bacterial enzyme. Say, for example, inhibitors of folic acid synthesis. Inhibitors of folic acid synthesis are para-aminobenzoic acid and pyridine. And pyridine synthesis is the enzyme which will act on para-aminobenzoic acid and pyridine, and they will produce dihydroteroic acid. Okay. Then further, dihydrofolate synthase will synthesize the dihydrofolic acid, and later on dihydrofolate reductase will ultimately give the tetrahydrofolate, and which will be the precursor or the raw material for thymidine, purine, and methionine synthesis. This is a normal mechanism which is happening in bacteria, but we have got certain antibiotics which is working at pyridine synthase level, that is sulfonamide. We have got trimethoprim, which is inhibiting the dihydrofolate reductase enzyme. And that's how the bacterial folic acid will not be synthesized. And if the bacterial folic acid is not synthesized, they will inhibit the synthesis of thymidine, purines, and methionine. And that's how the cellular metabolism will be stopped and bacteria will be ending its life. So that was about antibiotics and various classes of antibiotic based on their mechanism of action or site of action. Now let us talk about antifungals. Antifungals are the drug which is principally working on eukaryotic cells, because fungi are usually eukaryotes. They are having unique sterols in their cell walls, and we need to use the compounds which are congener of these sterols, and they prevent the cell wall synthesis. So that is principal mechanism behind all the antifungals. Now let me tell you about antivirals. Now here the challenge is little bit harder. Viruses are composed of nucleic acid, protein capsid, and host membrane containing virus protein. They really do not have their own cell machinery, and they rely primarily on host cell enzymes and host cell machineries. So if we want to prevent their replication, we actually will have to give a medication which will prevent the host cell physiology. Now that is also not good one, because in that case it will not, the drug will not be able to see which is the virally infected cell and which is non-infected cell. It will simply go and act on all the host cell metabolism, which is not acceptable. So we have targeted the antivirals which are typically binding with the viral metabolism and viral metabolites. And that's how we will be sparing the host to get adverse drug reaction. And some viruses have unique enzyme for DNA, RNA synthesis or protein cut, get those unique enzyme which will prevent the replication of the viruses. And additionally, what is required here for every antiviral to work is they should be able to penetrate the host cell. If they are not penetrating the host cell, then they won't be reaching to the target site, because viruses are usually where? Intracellular, within the host cell. So to prevent their growth and multiplication, the virus has to, or the antivirals has to go within the host cell. So that is about antivirals. Then coming to anti-protozoan drug, again the protozoa are eukaryotic cell, and we have got certain particular anti-protozoal drug which is targetedly working on protozoal metabolism only, and that is keeping the host unaltered. Anti-helminthic. Helminths are macroscopic multicellular metazoa eukaryotic organisms like tapeworm, roundworm, pinworm, and hookworm. We also have got specified anti-helminthic drug which targetedly work on the helminthic metabolism only, and that will not affect the host cell metabolism. Now, taste to guide the treatment, antimicrobial susceptibility testing, the commonly used and the older one is disc diffusion method, modified Kirby-Burr technique. I don't know whether you have learned it before or not, but it is simply done with the lone culture of the test bacterium on specialized medium called Mueller-Hilton agar. On Mueller-Hilton agar, to test the antibacterial susceptibility, we will first go for a lone culture of the test organism. Then we will apply particular antibiotic disc. Now this white color small disc which you see here on the plate are nothing but they are the particular disc for particular antibiotics, which is loaded with certain concentrations of antibiotics, and they will diffuse in the medium and they will inhibit the growth surrounding them. And we will check the growth after 24 hours, and if the growth is inhibited to a bigger zone, which you can see here in the first one top antibiotic disc, then that antibiotic is considered to be susceptible or sensitive for that particular bacteria. So that is how we detect the zone of inhibition of that particular antibiotic for that particular test organism, and that help us to identify which particular antibiotic is sensitive for the test organism and which particular antibiotic is resistance. If it is resistance, then you can just see here my cursor is moving. There is one antibiotic disc where surrounding them there is no growth is been inhibited. Can you see the white color thing which you can see here in entire plate is bacterial growth after 24 hours in lone culture. Okay, so this is the particular disc, and second disc is this one where you can see the growth is completely not inhibited and bacterium has grown very well. So those antibiotics are ineffective for that taste organism or resistant for that particular organism. There are other antibiotics like this one. You can see the zone of inhibition, which you can see here, a circle surrounding that antibiotic disc where the zone is created by inhibition of the growth of the bacteria. Larger the zone, more the sensitive antibiotic. But for this, we have got particular guideline with an organism and antibiotic combination. There are specified guidelines that if you get zone size larger than this diameter, then that is considered to be susceptible. Otherwise, it is resistance. So that will be taken care of while you are having the practical of antimicrobial agents. Two terminology which you should understand, that is MIC and MBC. MIC is minimum inhibitory concentration of one particular antibiotic for the said organism. If you are capable to achieve this minimum inhibitory concentration with that particular drug in patient's plasma and patient's body fluid, then that particular antibiotic will be effective and it would be killing the organism or inhibiting the organism. So MIC breakpoints are very important to be understood, and they need to be taken care well while you are giving antimicrobial therapy to the patient. While you are prescribing the doses, the MIC need to be taken care well. MBC is the minimum bactericidal concentration. That is the concentration at which one particular antibiotic will kill that particular organism. So that MBC is also need to be taken care well. There is another test which is known as epsilometry or epsilometer test, or in short, it is also known as E test. If you can see here the white color strip, there are three strips. These strips are having concentration of one particular antibiotic only, but in a gradient concentration. At the top here, wherever the 256 is written, it would be having 256 microgram concentration. But at the bottom, if you can observe here, the lowest point, there the concentration of the same antibiotic would be 0.16 microgram. Okay, so such kind of strips are commercially available. After lone culture of the bacteria, you can apply these strips and it will inhibit the growth of the organism in a cone shape. Okay, in a epsilon shape, you can see here a epsilon shape, and wherever the growth meets particular concentration, the inhibition will meet particular concentration. That concentration is considered to be minimum inhibitory concentration of that particular antibiotic for that test organism. So this is known as E test. Okay, now that was about antimicrobial testing. Okay, now let me tell you about resistance to antibiotic. Now the history is very much, evidence that whatever antibiotic we discover within next 5, 10, 15 or maximum 20 years, all the bacteria used to become resistant again. Okay, bacteria become resistant against those particular antibiotic, and they find out a bypass cellular pathway where the antibiotic cannot alter these bacteria, and they will keep on growing luxuriantly even in presence of these antibiotics. Okay, so bacteria are getting resistance. Behind that also there are certain cellular mechanisms. Okay, now which are these different cellular mechanism? One is blocking the entry of this antibiotic at porine channel level or cellular cell wall or cell membrane level, so that the antibiotic cannot reach to the cytoplasm, and the DNA synthesis inhibition or RNA synthesis inhibition or protein synthesis inhibition, whichever the target site for that particular antibiotic, the antibiotic will not be reaching there. And if the antibiotic is not reaching to the target site, definitely it would be ineffective. So that is one. Second is inactivating enzymes. Bacteria are very smart. They observe the compounds which we give as a antibiotic, and they start producing certain enzymes, which enzyme will go and turn inactivate or break down the antibiotic compound itself. And these enzymes are very well secreted outside the cell. Okay, then the third mechanism is altered target molecule or target site. Say for example, one particular antibiotic, example tetracycline, is effective or it is acting against 30 S subunit of bacteria where the protein synthesis is going on, and it has got a particular target site where the tetracycline attach and inhibit the protein synthesis of bacteria. But suddenly the bacteria changes that target site. They change the sequence of that particular protein by certain mutation, and they will alter the target site so that tetracycline, even if it has reached to the target site, it won't be attaching to that particular target site and they won't be effective later on. Okay, and the fourth one is efflux of the antibiotic. The fourth one is there is efflux pump mechanism, which will come continuously pour out the antibiotic from the cytoplasm. Whatever or whichever antibiotic has reached to cytoplasm, the efflux pump will continuously pull that antibiotic outside the cell membrane, and that's how they will not let the antibiotic to effectively inhibit any growth within the cytoplasm. So these are four important mechanisms by which the organism get resistance to. So what actually causes antimicrobial resistance? Which are the practices? Now, this is all what I have explained are the cellular mechanism of antimicrobial resistance. But you may have a query that how actually it is happening if we see to medical practice and if we see to prescription practice, if we see to rational or irrational use of antibiotics. It is this inappropriate antimicrobial usage. Do not use antibiotics wherever they are not indicated actually. Wherever, whenever and wherever they are not required, don't go for antibiotic treatment. Okay. Prescription not taken correctly. Number one, prescription is not taken correctly. Many people used to take excessive medication. Many people used to take that if my doctor has prescribed me 10 tablets, let me just take only 4 and later on I will see whether I need to take further or not. Okay, that is the most I mean inappropriate practice which leads to antimicrobial resistance. People are prescribing antibiotics left, right, and center, even if the clear indication of viral infections are there. Even if the patient is clearly suffering from common cold or flu, people will keep on prescribing the antibacterial drugs. Now, do you imagine or do you understand now after knowing the mechanism of antibacterial and antiviral that these are of no use in case of common flu or viral infections? Okay, so that is overuse. Another thing, antibiotics sold without medical supervision. The pharmacist, if you go to a pharmacist, and I'm telling you current day situation, if you go to any pharmacist or a private pharmacy and you ask them that I'm feeling cold like symptoms, sore throat, and had a fever, he would have prepared one packet with azithromycin, paracetamol, and all of the required medications, and they'll simply hand that over packet to you. Just give me 200 rupees. That's over. Now that kind of practices are again keeping antimicrobials in the category of something I would, in a layman term, I would call it as mukwas. People are consuming the antibiotics something like mukwas. Chalo, I had this allergy and I just had exposure of the dust uh, today and I've started having symptoms. Now in allergy, role of antibiotic is nowhere and people will start taking antibiotic even if there is allergic attack. So these practices are the worst practices. And spread of the resistance microbe in the hospital due to the lack of hygiene and poor infection control practices. That is again a biggest risk which will generate a superbug or multi-drug resistant organism in a hospital acquired flora. Lack of quality control in manufacturing of and outdated antimicrobials are sold without any, I mean, ethical concern. Okay. Inadequate surveillance or defective susceptibility assays, inadequate antimicrobial culture testing, and inadequate or defective susceptibility testing by the laboratories. Either physicians do not send the culture samples whenever even it is indicated, and without sending the culture sample, they will simply ask the patient, if I'll send the culture sample, that will cost you around 1500. What do you want to do? Whether you want to go for the test or you want to have antibiotics of 100 or 200 rupees and just get yourself cured, then the patient will definitely prefer to have cost effective option and they will definitely be happy without doing culture. But this is unawareness. Without taking investigation, if you rationally or irrationally prescribe the antibiotic, it is of, I mean, deadlier outcome in future. Poverty or war, which will lead to lot many infections spreading easily and rampant. Okay, use of antibiotics in food, particularly animal food, is also of great danger to the mankind. Okay, and just to show you the data of very old time, 1994 1997, tuberculosis resistance was very less across the globe. But if I'm telling you the situation of today, tuberculosis resistance is now very common across the globe. Wherever there is tuberculosis resistance to anti-tubercular is very rampant. You can see the picture of 2012. Okay, just compare both the image. The one picture is of 1994 to 1997 data, and another picture is of 2012 data. No one is spared. Simply no one is spared from the anti-tubercular drug resistance. Consequences of antimicrobial resistance. Infections resistant to available antibiotics, and infection cannot be treated, and the people die because of scarcity of antibiotic, and particularly sensitive antibiotic. And increased cost of the treatment, lengthening of the hospital stay, and lengthening of the complication list for the patient. Okay. And you can also see this data: cost of treating multi-drug resistant TB. First line is very less, second line is very high, and third line is even beyond second line. So you can just imagine the figures which is indicated in US dollar. They goes very high if you have anti-tubercular resistant tuberculosis infection. The coming cost of superbug, 10 million deaths per year. This has been forecasted, and in 2050 the deaths of individual area would be something like these. In the map of globe, you can see the number. This is just a forecasted figure, and if we will not control ourselves for prescribing proper antibiotics, the figures could be even scarier than this what we see. These are figures of death per year, number of deaths in the population per year. Simply understand the severity. What factors promote antimicrobial resistance? Exposure to suboptimal level of antibiotic, that is one. The case which I have told you before, that physician have prescribed 10 tablets, but the patient will decide on his own that I'll take only 4. And if I'm okay at the end of the 4, I'm feeling good, then I'll stop taking another 6 antibiotic. Now this is the most poor practice. Okay. And another thing is exposure to microbes carrying resistance gene. If the normal flora is exposed to a resistant flora which is carrying a very resistance gene through plasmid or anything else, then your normal flora which was susceptible before will now turn to resistant one. And genetic mutation causes the drug resistance. A non-resistant bacteria exists. Bacteria multiply by binary fission. Some mutations make the bacterium drug resistant, and now the drug resistant bacteria used to thrive and exceed. There are other mechanisms of acquiring drug resistance, genetic transfer of the drug resistant, that conjugation, transduction, and natural transformation. Conjugation is nothing but it is happening in the bacteria which has got capacity to produce pilus. One bacteria which has got plasmid will have a bridge in the form of pilus with another bacteria which is not having plasmid for the drug resistance, and it will make a copy of that plasmid and it will donate that plasmid to sensitive bacteria, and that's how the sensitive bacteria will become resistant. Okay. Transduction is another one, where the transduction, a faulty bacteriophage packing, faulty nucleic acid packing in the bacteriophage will transmit the drug resistant gene to a sensitive variety of bacteria and ultimately convert the sensitive bacteria into resistant. Natural transformation, in that the resistant donor cell will be dying and its nucleic acid will be surviving as a free DNA in the environment, and this free DNA is taken up by another sensitive bacteria, and now the sensitive bacteria has become resistant. So this is how the genetic transfer of the resistant mechanism also occurs. Okay. A proposal to combat the antimicrobial resistance. Now what are the ways to handle this problem? Speed development of new antibiotic, track the resistant data nationwide and restrict the antimicrobial usage, and direct observed dosing which has been implemented in tuberculosis that can help us to monitor well. Okay, use of more narrow spectrum antibiotics and use of antimicrobial cocktails to treat the bacteria harder. WHO has also given one statement in 2013, antibiotics should not be used after surgery. Okay, now that is also again not practiced well. During surgery also people practice antimicrobial therapy rampantly, and they irrationally give the higher antibiotic before the surgery, during surgery, and even after the surgery. Okay, so that has to be stopped, right? And now, how to combat the resistance? And as I have mentioned before, in 2018 there was antimicrobial resistance awareness week, which was celebrated by WHO. At the end of that 2018 celebration, they had a convention, and during this convention, and multiple authorities advice and recommendation, they came out with one program that is known as antibiotic stewardship program. Now, antibiotic stewardship program need to be implemented at institutional level, nursing home level, low resource hospital level, or critical care unit level, and OPD patient level, because antibiotic stewardship program are having different structure. If it is institutional, if it is nursing home specific, if it is OPD patient specific, the module will be changing. So based on the structure where you are fitting into, the module can be implemented. Now, how the program works, I'm just giving you a brief idea about it. There could be three variety of the program. There could be having there we could have a broad program, we could have a pharmacy driven antibiotic stewardship, or we could have an infection and syndrome specific stewardship. A broad program will be a general program which will just restrict the use of higher antibiotic if it is possible, case to case basis. But that is very difficult to achieve, and the standards to meet the standard is also very, I mean, tough. So pharmacy driven is another thing which is possible in small nursing care units or hospital or institutes, where selling of the higher antibiotic need to be restricted and it can be controlled, something like narcotics. Okay, and it is issued after some pre-authentication and some, I mean, some authorities permission only. It is not freely sold to everybody, even in spite of prescription. That is pharmacy driven. You can also record and report the consumption of the broad spectrum and narrow spectrum antibiotics month to month. And whenever you see rise in consumption of the broad spectrum antibiotic in one particular month compared to the previous average, you can directly trace the bills and you can contact that particular physician and you can counsel the physician to control the prescription. Okay. The third and the most effective way of implementing the antibiotic stewardship is infection and syndrome specific stewardship, where we classify the infections in either community acquired infection, hospital acquired infection, or some syndrome like UTI, urinary tract infection, septicemia, pneumonia, surgical site infection, wound infection kind of syndrome. We can divide, and for that particular syndrome only, we, with the consent of the hospital management and the clinicians, we can devise a document or we can generate a document where we will advise the people to prescribe only specific antibiotic in specific conditions. Observe the response for another 24 to 48 hours. And if you do not get response in the patient, and if the patient do not improve, just increase or just go for higher antibiotic or escalate to the higher antibiotic based on the culture and susceptibility report. And these are the core elements who are the party to be implementing this antibiotic stewardship program. The hospital leadership, first of all, should understand the importance of this antibiotic stewardship program. They should understand the gravity and they should take a leadership role in implementing. They can divide a key support team, which could be a second layer below the hospital leadership team. They could be clinicians, they could be pharmacists, they could be critical care intensivists, they could be microbiologists, they could be infection control nurses for audit and various operational element. Every individual who is identified as key support team should be counseled for the accountability and the responsibility in the entire program. The pharmacy is the place from where you will audit and you will trace the billing and issue of this antibiotic, and you will monitor the consumption of the antibiotic usually. And what are the actions to be taken? Action is local antibiogram for your particular hospital has to be prepared, looking to the antimicrobial susceptibility data of last 1 year or 2 year, and they have to be reviewed every 6 monthly or 3 monthly based on the convenience. Prescription policy has also to be properly done, and the prescription has to be audited well by the infection control nurse, microbiologist, and pharmacist. That some consultant, if not following the policy, then he or she has to be counseled well to bring the correct practice in place. And pre-authorization of the, what can we say, is the prescription. Say, for example, if somebody is prescribing a very higher antibiotic like colistin, one has to get the permission from management or some authority like microbiologist or somebody else who is looking after the entire stewardship program, and tracking of the bills, recording and reporting of the consumption and audits, and education of the healthcare worker for a judicious use of antibiotics. So these are the core elements, and misuse of the antibiotic puts us all at the risk, and we may end the pipeline for our next generation to come. And for our next generation, they may not have any antibiotic. Think twice, seek advice before consuming and prescribing antibiotics. Take the advice from an authorized physician only. So that's it for antibiotic stewardship, antimicrobial sensitivity testing, and antimicrobial compounds. Okay, if there is any query, we can discuss, or else I will conclude this lecture. Please respond. If there is any query, we can wait.